This paper highlights the importance and structure of the coronavirus spike protein in the transmission mechanism of the virus, and thus a way therapeutics could be designed to target the virus. Generally speaking, this paper was able to isolate two neutralizing antibodies against SARS-CoV-1 and MERS-CoV, and determine the mechanism of their neutralization. Before my colleagues explain how they reached these results and their importance, I'm going to start with background on the structure of the antibodies used and the coronavirus. So here you can see a very simplified structure of a traditional IgG antibody. Antibodies are proteins produced by plasma cells that are used to neutralize pathogens and viruses. The Fab region is the antigen binding fragment and is the region of the antibody which binds to the antigen. In this case, the antigen is SARS-CoV-2. The Fc portion is responsible for the biological activity. This study used llama antibodies, which is important to note because camelids have heavy chain-only antibodies which contain a single variable domain. Once the Fc region has been removed, these are referred to as single domain antibodies, or VHHs, also known as nanobodies, which can be seen in the image to the most right. In this state, they can have affinities and specificities for antigens comparable to conventional antibodies. There are several reasons why VHHs have proved advantageous as therapeutics for respiratory disease in particular. One is their enhanced thermostability and chemostability in their multivalent or bivalent form. Also, they've already been used as a therapeutic in a common respiratory disease known as RSV. Additionally, highly stable VHHs can be nebulized and administered via inhaler directly to the site of their infection in their lungs. Their ability to maintain their stability for long-term storage would allow these therapeutics to be stockpiled and makes them an ideal candidate for a treatment option in case of an epidemic like the current COVID-19 crisis. It's important to understand the structural basis of coronavirus to understand its transmission mechanism and subsequently how therapeutics must be used to target the virus. Coronaviruses are single-stranded positive-sense RNA viruses. The structure of coronaviruses are covered in spike glycoproteins, often denoted with an S. The spike protein is responsible for engaging the host cell receptors and catalyzing membrane fusion between the lipid membrane of the host and lipid membrane of the virus. This process allows the viral genome to enter the host cell and begin the process of replication. The S protein is a trimeric complex with two distinct subunits. S2 contains the fusion machinery, and S1 contains the RBD, or the receptor binding domain. These receptor binding domains are the main targets for the VHH antibody therapeutics mentioned earlier and key in serving the aforementioned functions of the spike protein. These RBD are responsible for binding to host receptors. The host cell receptor that engage are ACE2 for SARS-CoV-1 and DPP for MERS-CoV. SARS-CoV-2 also uses ACE2. The receptor binding domains can be seen in two functional conformations, as noted in figure 5a. In the down conformation, the receptor binding domain masks susceptible epitopes from the human immune response, thus protecting the virus from neutralizing antibodies. In the up conformation, it's accessible for receptor engagement. It is hypothesized that the receptor binding event would trap the receptor binding domain in the less stable up conformation, leading to the gradual destabilization of the spike protein until it is finally triggered to initiate membrane fusion. Many of the current anti-SARS-CoV-1 RBD and the anti-MERS-CoV RBD antibodies which have been identified have a mechanism which blocks the receptor binding site in order to trap the receptor binding domain in its unstable up conformation. These proteins are a prime target for therapeutics as they play a key role in the transmission of the virus.
So this experiment can be split up into three main groups, and that begins with the isolation of single domain antibodies that could potentially neutralize MERS-CoV and SARS-CoV-1 S proteins. Additionally, they wanted to map the epitopes targeted by these isolated antibodies and then solve the crystal structure of the interaction between the two. Finally, they wanted to test the isolated antibodies to see if they were capable of neutralizing SARS-CoV-2 as well. In other words, this is the virus that causes COVID-19. So this begins with the serial immunization of SARS-CoV-1 S and MERS-CoV S proteins in a single llama. From there, they purified and assembled a library of antibodies, and after 2 rounds of biopanning with phage helpers, they were able to select for positive clones. Those clones were then sent through multiple sequence alignment and a phylogenetic analysis, and that's where they revealed that there were 7 unique antibodies specific to MERS and 5 unique antibodies specific to SARS. Then they quantified the binding affinity of these antibodies using an enzyme-linked immunoabsorbent assay. So these are the results from the ELISA. They're split up into 2 graphs based on their respective viruses. Looking at the MERS side, antibodies of interest are antibodies 55, 12, 34, and 40. And for SARS, those antibodies are 72, 1, and 6. And the importance of these are that they show the highest binding affinity to the S protein. Additionally, the group wanted to see if these antibodies were capable of neutralizing their respective viruses. So in order to do so, they created pseudotype viruses by transfecting T93 T cells with a plasmid that encodes a luciferase reporter, a lentivirus backbone, and of course the spike proteins of each individual virus. They mixed these with serial diluted antibodies, and then they measured the relative luciferase activity. This is important because the viral S protein is fused to the luciferase reporter gene. So any luciferase activity measured is directly correlated to the viral activity. But the percent neutralization was calculated to where no virus in the sample would result in a 100% neutralization, and no antibody in a completely viral sample would result in a 0% neutralization. And then after that, they sequenced the neutralizing antibodies. So here are the results of the neutralization assay. What you're looking at in these values is the lowest number. Essentially, these values represent the concentration required from each antibody to neutralize at least 50% of the virus. So the antibodies of interest here are MERS 55 and SARS 72. They show the lowest concentration, meaning that they have the highest neutralization ability. Additionally, all antibodies capable of any sort of neutralization were sequenced, and when you compare the complementarity determining regions, the CDR regions essentially, when you look at their sequences, and especially for MERS, you look at all the antibodies, they show little variance in their sequences, and this essentially means that they're more likely to bind to the same epitope. Conversely, when you look at the SARS antibodies, they show a lot of divergence in their sequences, and this means that they are less likely to bind to the same epitope.
Once the antibodies were isolated, the second part of the experiment was to map epitopes and determine the crystal structures of the interactions. This slide is an overview of the different steps they took to accomplish that. The first step was to map the epitopes targeted by the VHHs. MERS S1 RBD and N-terminal domains, as well as SARS1 RBD and NTDs, were tested with their respective VHHs using ELISA. The graphs show that all MERS VHHs bound to MERS S1 and RBD in concentration-dependent manner. SARS VHH72 strongly bound to the RBD. Neither MERS or SARS VHH is bound to NTD, as can be seen in the bottom left and right-hand graphs. Once epitopes were found, the team used SPR to find the binding kinetics and affinities of the interactions. MERS had a KD of 79.2 picomolar, which is partially caused by the small off-rate constant. The SARS interaction had a KD of 1.2 nanomolar. With the epitopes mapped and the kinetics determined, the next goal was to solve the crystal structure of the interaction. MERS VHH55 and RBD complex showed the extensive binding of VHH's CDRs with MERS RBD. Most importantly, CDR3 of the VHH looped over the DPP4 binding interface, as can be seen in Figure A, which includes DPP4 from productively engaging the MERS-CoV RBD, as can be seen in Figure B. Further analysis of the crystal structure shows that the network of interactions from the CDRs and RBD suggests that RBD residue arginine 542 has a critical role in MERS VHH55 binding, as can be seen in Figure C and D. That arginine was also previously identified as a conserved residue crucial for high affinity of DPP4 binding. Given that the MERS interaction had such a high affinity of 79 picomolar, the team wanted to investigate the reason. To do this, phenylalanine 506 was substituted with leucine. This residue was chosen because it is a natural variation of that position in various MERS strains, and the rest of the RBD is highly conserved. SPR was used to find the affinity of the leucine substituted RBD with MERS VHH55. It was determined that it reduced the binding affinity by 200 times to 16.5 nanomolar. The team concluded that VHH55 would be able to recognize various MERS strains, but was not likely to cross-react with other distantly related spike proteins. With the MERS structure resolved, the crystal structure of SARS1 RBD and SARS VHH72 was investigated. It was determined that CDR2 and 3 bear significant areas of the binding interface, but the epitope does not overlap with the ACE2 footprint on the SARS1 RBD, as can be seen in Figure 1. This shows that the ACE2 receptor clashes with the antibodies framework, as can be seen in Figure B and C. Further, ACE2 has an N-glycan modification at asparagine 322 that clashes with VHH72 when ACE2 and SARS1 RBD are bound. The high affinity of VHH72 and SARS1 RBD can be accounted for due to the network of hydrogen bonds created with VHH72's CDR 2 and 3 and the RBD domain, as can be seen in figures D and E.
So after finding the crystal structure of the binding interaction between SARS VHH72 and SARS-CoV-1, RAP's research team wanted to investigate whether or not VHH72 can cross-react to SARS-CoV-2. In other words, can VHH72 bind to CoV-2 and not just CoV-1? And this is because, as you see in figure B, SARS-CoV-2 is very similar to SARS-CoV-1, and SARS-CoV-2 is what causes coronavirus 19. And so if you see in figure 1, using surface plasmon resonance, scientists found that in fact VHH72 has a very high binding affinity to SARS-CoV-2. And it had a binding constant of around 40 nanomolars, which was a good amount higher than the binding constant for CoV-1 and VHH72, which was around 1.2 nanomolars. So a difference from about 1 to 40 nanomolars means that CoV-2 would have VHH72 fall off at a little bit higher of a rate than if it were bound to CoV-1. And we'll come back to that later. So after the researchers figured, okay, well, VHH72 can in fact bind to CoV-2, what's going on? What is happening in these coronaviruses when it does bind? So if you take figure A as an example, in MERS, VHH55 can bind to the down conformation of a spike monomer out of 3 different monomers. And so when it binds to this down conformation monomer, if any of the 2 neighboring monomers are positioned in the up conformation, then they will have this steric clash with this VHH55 shown in this red circle. This steric clash that prevents the up conformation monomer from transitioning into a down state. And if you think about a live human cell that gets infected by any coronavirus virus, what happens is an up conformation monomer will recognize or be recognized by a cell membrane receptor. And then it will be locked in that position. And after a while, these other little monomers will transition from downstate to upstate. And overall, this coronavirus will transition into from a pre-fusion state to a post-fusion state and then fuse with the cell membrane. And so what happens is when this antibody attaches here, it mimics the cell membrane receptor and it forces this S-spike protein to be up, this S-spike monomer to be up. And then the other monomers eventually trigger up and it triggers this transition into the post-fusion state without actually having met a cell. So basically, VHH triggers this usage of this virus without actually having it infect the cell. And so this was what happened in SARS-CoV-1 and also SARS-CoV-2. Scientists also thought there could be an extra mechanism for neutralizing these viruses. And what they did was they used a BLI-based assay in which they fixed coronaviruses onto a plate, and they dipped it first into an antibody solution and then into a solution with a cell receptor such as DPP4 or ACE2. And what they found was that in the cell lines that were dipped into antibody first and then into the well containing the cell receptor, there's very little response, very little activity, because basically the coronavirus was not able to interact with the cell receptor. And so maybe VHHs can prevent their ribosomal receptor binding domains from even interacting with the host cell receptors. So this would seem like VHH is a very good treatment option in general by itself, but there is a complication, which is that, as I said earlier, despite the high binding affinity of VHHs to the virus RBDs, there's actually a higher dissociation rate than in CoV-2 compared to CoV-1, which prevented the VHH72 from neutralizing the virus successfully. So in other words, the viruses had VHH fall off too quickly for them to be neutralized. So RAP's research team got around this kind of cleverly. They produced 2 bivalent SARS VHH72 strands, and one of them, as an example, this first one over here, VHH72, VHH72, had basically 2 VHHs joined back to back, so that you have 2 of them that could both potentially interact with the coronavirus. So there's a double chance for it to stay onto the coronavirus rather than falling off. And then the researchers also created another one where the VHH72 was genetically fused to human IgG1. And so these 2 could stick to the coronavirus better. And this successfully compensated for this high rate of dissociation of the monovalent SARS VHH72. Like bivalent could stick on better. And also, the bivalent ones could express pretty well in transplanted cells, and moreover, they were successfully able to neutralize these SARS-CoV-2 pseudoviruses in vitro in class 2. So overall, I just wanted to go over what we did in this experiment, or what the RAP did in this experiment. They isolated the single-body antibodies from llamas, these VHHs, and they could cross-reactively neutralize both MERS-CoV and SARS-CoV-1. So the researchers figured out the epitopes targeted by these VHHs, and they solved the binding interactions by solving the crystal structures. And then they eventually designed bivalent VHHs that could neutralize SARS-CoV-2 in vitro. These are treatment. So for future research, researchers could investigate like a nebulized treatment in which this bivalent VHH72 is basically sprayed into the human lungs, kind of just a spray that's inhaled. And that's because SARS-CoV-2, the virus that causes COVID-19, likes to bind to human cells there and cause COVID-19. So VHH72 will beat it to the punch and neutralize SARS-CoV-2 before it can do any damage. And so I just want to go through the references. These are references, and that's about all. Thank you very much.